Hello, everybody. My name's Matt Vanderheiden, and I'm going to be your moderator for the morning session. So let's move right on to the speakers. Our first speaker is really someone who needs no introduction. It's Bert Vogelstein, who's the director of the Ludwig Center at Johns Hopkins. He's a Lustgarten Foundation Distinguished Scholar and an investigator of the Howard Hughes Medical Institute. As all of you know, Bert has been a real leader in understanding the genetic basis of human cancer, and today he's going to talk to us about cancer genomes and their implications for prevention. Thank you very much for coming.
Thank you. I'm really delighted to be here. This is a very special conference in my view. It's the first one that I know of, a general scientific conference that's devoted to prevention and early detection rather than a specialty conference. And it's particularly meaningful to me because I believe that prevention and early detection, as Ed and Nancy just said, are not only extremely important, but perhaps the most undervalued, underfunded, and under-researched components of cancer investigations today. And what I'll talk about, I changed the title a little bit to somatic mutations and earlier detection. And I want to emphasize earlier. You'll see why in a few minutes. So I'll divide my time into three parts. The first part is a discussion of what causes the mutations. The second part, how heterogeneous they are. And the third, how they can be exploited. And each of these parts has important implications, I think, for the way we look at prevention and early detection. So if you look at the, say, American Cancer Society website or many others, you'll see this kind of thing. What causes these mutations? There are two causes. One's environmental. The other is hereditary. But that's actually only part of the story. And here I'll emphasize the work of Christian Tomasetti, who's a mathematician now at Hopkins. So what Christian discovered, in a sense, and I'll present this in a different way than the actual description of the observation, but I hope it will make it clear. Suppose one were to do autopsies in 1 million people in the United States, totally normal people when they died, other than what they died from, of course, and looked at the concentration of a compound, let's call it compound R, in their normal tissues. Suppose one had found a rather striking correlation between the concentration of that compound in their tissues and the rate of cancer incidence in those same tissues. For example, the concentration of R would be high in colorectal cancers, which are very common, and low in bone cancers, which are much less common. Suppose, furthermore, one discovered that this correlation was extremely statistically significant, and also that this newly discovered compound R is a potent mutagen. Well, the obvious implication of those results for any scientist would be that compound R plays a very major role in cancer incidence. That's the most obvious explanation, and there are really few others that one could posit that even come close to its plausibility. Now, what Christian discovered was that it's not that compound R, so to speak, is just cell division. If you look at the total number of cell divisions in a tissue over a lifetime, that's very highly correlated with the incidence of cancer in those tissues. And remember, every time a cell divides, whether it's a stem cell or any other cell, approximately 3 new mutations occur. So cell divisions is a potent mutagen. Now, the implications of those observations I told you the observation itself. This is an interpretation of that observation that Christian and I made, were that about roughly, say, 2/3, something like that, of the mutations that occur in many cancers are due to this R factor, simply replicative mutations that occur during normal cellular processes, with the rest due to the combination of environment and heredity. Now, why is this important for prevention? Well, let's imagine a theoretical scenario. One of the purposes of this symposium, I believe, is to think futuristically. So let's imagine that we destroy planet A. It doesn't take much imagination. So 5,000 years from now, we all have to move to planet B. Well, let's pick one that has no ultraviolet radiation from its suns, and we eliminate all carcinogens, industrial or natural. And furthermore, we engineer cells so to remove all hereditary causes of cancer. That'll be easy in 5,000 years. You just go to the local drugstore, get your CRISPR package, do whatever you want. So on this theoretical planet, will there be any cancers left? And the answer is yes, because there's no way, at least easy way, to eliminate the other source of mutations. Now, cancer incidence will certainly be reduced, but it won't disappear. Fortunately, secondary prevention, which Nancy defined in her remarks, can still be practiced and still limit cancer deaths on planet B if we devote more energy so that in 5,000 years from now, we will have better secondary prevention, that is, early detection methodologies. And as Ed mentioned, if research continues on the same path as today, that may not happen, hence this symposium. Another way that these observations are related to prevention is one can ask, are these replicative mutations that occur during normal stem cell divisions truly unavoidable? Well, look at the causes of endogenous mutations. There have been 4 that have been determined by molecular biologists so far. There's an interesting paper last year in Nature on the quantum effects on base pairing. Certainly, that would seem completely unavoidable. Mistakes made by polymerases are engineered through evolution. Our cells make mistakes because, obviously, that drives evolution. Cancer, in one sense, is simply a side effect of evolution. Hydrolytic deamination of bases occurs and is a source of mutations, as is damage from endogenous reactive oxygen species. These are reactive oxygen species that are produced as a result of normal respiration in the mitochondria. We can't get away from them. It's not due to exogenous sources of ROS. Now, we don't really know what fraction of the mutations are due to each of these 4 factors, but at least we can imagine that some of them, maybe a major portion of them, are actually preventable through some sort of chemoprevention, kind of the statins for cancer, if you will. If one could discover drugs that reduced endogenous mutations, such as antioxidants, not a new concept, but hasn't really yet been integrated into a genetic context, then perhaps we could reduce some of these. Okay, let me go on to the second point I'd like to make, which is about the heterogeneity of the somatic mutations in cancer. So, Heterogeneity is an oft-used concept. It's difficult to find a paper today that doesn't have heterogeneity in its title if it discusses cancer somatic mutations. But that word heterogeneity has little meaning unless it's quantified. Big or small doesn't really mean much unless you put a unit on it and compare it to other things that might be bigger or smaller. So what is the genetic heterogeneity among totally normal cells? For example, these two Marines are obviously identical twins. Suppose you were to sequence cells from one of them and compare it to the other. You would find they are far from identical. There are thousands of mutations in any two skin cells that distinguish one twin from another. Now, you can apply the same sort of analysis to a single individual. Let's take a randomly selected one. And let's do a brain biopsy and pretend we could find two neurons. I'm having much too good a time here. So what would we find? We would find they're far from identical. They have thousands of mutations that distinguish any two cells in the brain or any other organ. And Martin Nowak's team, which includes Ivana Bozic, Hannes Reuter, and Jeffrey Gerald over across the street at Harvard, have looked at this quantitatively. And they find, actually, that the genetic heterogeneity among cancers is quite a bit less than in normal tissues, because cancers, of course, are characterized by bottlenecks in evolution that, in a sense, erase heterogeneity. The point I want to make, though, is not that there is no heterogeneity. There is some heterogeneity, and some of it has great clinical importance. For example, here is a typical patient on a typical clinical trial of advanced disease. You can see all these metastases. Now, drug resistance has its origins in a specific type of heterogeneity. That is, the heterogeneity within the metastatic lesions that are being treated. These are patients that have failed surgery, and now their only hope is systemic chemotherapy, targeted or otherwise. The fundamental basis of drug resistance is that in any cell, again, this is Martin Nowak's team's work, in any metastatic cancer cell or cancer lesion metastases, there are hundreds to thousands of resistant cells already present in each metastatic lesion prior to therapy. These aren't induced by therapy. They can be, but most of them are present prior to therapy, limiting success and explaining why recurrences, even when the targeted therapies work, occur within a few months to perhaps a year after targeted therapies are administered. Now, this is relevant to earlier detection, and let me show you why. We are all good at curing mice. I am particularly good at it. If you are a mouse and someone has rudely injected a tumor into you, come to my lab. I can offer you hope. Unfortunately, I have never cured a human being. Now, why are mice easier to cure than humans? Some might say, well, they're artificial models. I don't think that's it. I'm enormously impressed with the sophisticated murine models that have been developed over the last 20 years, both here at MIT and many other places. I think the explanation is much simpler. Let's look again at a typical patient with cancer. Lots of big metastatic lesions, tens to hundreds of billions of cells. Now, let's compare that to a mouse. Here's a whole mouse. There's a large mouse tumor, actually too large to be allowed by the veterinarians in charge of mice facilities. It's orders of magnitude smaller, which means there are orders of magnitude less cells which have the potential to become resistant. And in order to have confidence that one is going to cure a human with a targeted drug, the preclinical data should show that it cures cancer in every one of 100 mice. That's the challenge. And very few studies, preclinical studies, meet that bar. But that's really what you need. Now, one of the goals of earlier detection is to make tumors, human tumors, more like those of mice. We don't have to necessarily detect tumors before they have metastasized in order to make a difference in patients' lives. We can simply detect them before those metastases have grown to these very large lesions, and then we will have as good a chance, or at least a better chance, of curing them with whatever therapy is administered. And this point is extremely important because sometimes the fields of prevention and early detection are looked at as, in a way, opposed to therapy. They're not. They're intimately linked. Whatever form of therapy you have, whether it's standard chemotherapy, targeted therapy, immunotherapy, any new therapy you can design, that therapy will work better in mice than people, and it will work better in people who have less disease. So these fields should be integrated, not contrasted. And in my view, one of the best things industry could do is help in this effort because it will make their drugs work much better. Finally, the last topic. How much time do I have left, Tyler? 5 minutes? Okay. I'll quickly go through this. How can these somatic mutations be used for earlier detection? This is our current model of cancer, 3 steps: initiation, expansion, and finally metastasis. From the viewpoint of early detection, the most important impact of this model is that it takes 2 to 3 decades to develop most cancers. That provides a large window of opportunity for early detection and early prevention. Cancer does not develop overnight. It's a long process. Now, looked at in a patient or person, human-oriented fashion, all these people at a typical football game, they represent the number of people who will die from colorectal cancer in North America today. Every one of those people who will die from colorectal cancer will only die because their tumors were not detected in the first 27 years of the tumor's existence. Obviously, I'm rounding. An enormous opportunity for intervention and a large window in which to do so. And one of the approaches for earlier detection that our group has long been involved in is the detection of driver gene mutations themselves. And this has the obvious advantage of exquisite specificity. Conventional markers are not really specific for cancers, nor are they pathogenically related, whereas genetic alterations, as we all know, are the proximate causes of neoplasia. The technology we used was developed by Isaac Kindy, a very talented MD PhD student in our laboratory. It was published in PNAS a few years ago, and it's best called molecular barcoding. Each template molecule is given its own barcode, and this reduces errors in sequencing by roughly 2 orders of magnitude, which is adequate for the task. And Isaac, together with Yuxuan Wang, another MD PhD student in our lab, and other faculty members, have looked at this in various ways. So here's a result that was published in Science Translational Medicine a couple of years ago. The orange bars represent relatively advanced tumors, stage 3. Now, this can't be seen as very early detection, but it's earlier detection for sure. And let me show you some new data just derived by Jeannie Tai and Peter Gibbs in Melbourne in collaboration with Yuxuan at Hopkins. What they did is look at stage 2 patients who generally are not treated with chemotherapy and tried to evaluate whether circulated tumor DNA, ctDNA, could be found in some of those patients after surgery and in time for therapy, adjuvant therapy. And what they found is shown here. You can see quite clearly that 4 weeks after surgery, when there was no evidence, either clinical or radiologic, of metastatic disease, many patients already had it because their primary tumors were removed, but you could see circulating tumor DNA. The window here is quite large, with a risk of almost 18-fold. And this kind of study opens a lot of opportunities, especially for pharmaceutical companies. Most drug trials are now done on advanced cancer patients, but in the future, I hope they can be done, or more of them can be done, on earlier stage patients. The problem is they have no disease to follow. So you can't tell whether the drug is working until you study 5,000 patients for 10 years. Very difficult to do adjuvant trials. This could make it much easier because you can focus on those which already have occult metastatic disease and see if you can get rid of it. The other part of this slide is in early stage patients, indicated by the red bars, and various tumor types on the bottom, 4 or 5 common cancer types. And here, the detectability of circulating tumor DNA ranges from 30 to 70%. It depends on the tumor type. Now, this is the maximum detectable that you can get because it's biologically limited. In this study, what Isaac and Chaitin did was to look at a mutation in a tumor, a driver gene mutation, and then follow that mutation in the blood of these patients. The sensitivity was maximal. They could detect 1 mutant molecule in 10 mils of plasma, 20 mils of blood. So what's limiting here, this is an absolute ceiling, I think, of what you can detect with circulating tumor DNA or liquid biopsies. Now, this is a more realistic liquid biopsy, so to speak, circulating tumor DNA in real patients, which Yuxuan has started. So far, she's looked at 220 cancer patients and found a little more than 40% of them are detectable within that ceiling using a multiplex PCR-based assay based on Isaac's technology. As would be expected, false positive rates are very low, less than 1%. In terms of staging, also as expected, the only tumor type she studied in which enough early stage patients have been enrolled are ovarian cancers, and it's close to 50% in more advanced ovarian cancers and in the 30s for early stage. Now, how do we look at this? 30 to 40% is not 100%. It's not what we'd like, but it's a start, and it's kind of like the glass half full or half empty. If you developed a new drug that could potentially cure 30 to 40% of cancer patients, you would make billions of dollars, but more importantly, everyone would appreciate the significance of that. With early detection technologies, it offers the same hope for reducing cancer deaths, but it hardly makes any dent on the way people think about its importance. And that's the one thing we need to change. And I'll close in the last few slides with potential ways to increase that sensitivity. And one such way is to combine circulating tumor DNA with released tumor DNA in bodily compartments. And I'll give you one example. This is pap smears. The same pap smear DNA that can be used for HPV detection can actually be used for detecting endometrial or ovarian cancers. But not by looking for HPV, of course, by looking for somatic mutations. And here, again, is some recently as yet published data from Yuxuan. In endometrial cancers, which still unfortunately kill a lot of women in the United States today, you can detect 93%, and it doesn't matter whether they're early or late. With ovarian cancers, you can detect about 40% by looking at the pap smear fluid. If you combine that with circulating tumor DNA, you get up to a pretty respectable 70% or so, which is pretty equivalent to what you can do with mammography or what you can do with HPV testing for or conventional conventional pap smears. So it's in the range where it starts to be attractive clinically. At any rate, there are many other approaches. I'm agnostic about which one. We work on somatic mutations because that's our experimental expertise, but that's not necessarily the best technology. One can think about epigenetic changes, cancer cells themselves, transcripts, metabolites, proteins, and my favorite is cancer-specific imaging features, molecular imaging, so to speak. At any rate, the point I want to leave you with is, again, futuristic. 50 years from now, we'd all like to be able to reduce cancer deaths by 3/4. I think that's feasible, but only if we implement earlier detection, more prevention strategies, as well as design new therapies. These things all go hand in hand. Finally, I'd like to thank the members of my lab, particularly Ken Kinsler, who co-directs it, and disclose and acknowledge the various companies that we are working with to help make some of these tests commercially viable. Thank you.